Okay, cool. Thanks. This is Onur. I'm a grad student in Professor Saif's lab, and I've been an EBICS trainee for about 5 years now, working on biohybrid machines. So I'll start briefly with a discussion of what we've done so far, what we want to do, and how I think we might be able to go about achieving our goals. So this is just a background slide showing various muscle-powered biohybrid machines or biobots that are capable of locomotion, motile bots, walking or swimming. Many of these examples came from EBICS groups. And what I've been interested in when I started my PhD was to integrate neuronal control on top of this, because in nature, animals that use muscle to generate movement and locomotion coordinate the activity of their muscles through the nervous system. And the first step for introducing neuronal control is forming neuromuscular junctions. The neuromuscular junction is basically the interface between muscle and the rest of the nervous system. And the key cell type here is the motor neuron. So this is the swimmer that we developed and published last year. We basically developed a technique for doing a neuron muscle co-culture on a freestanding scaffold. So this was a sort of engineering or craftsmanship challenge. Most of the biological aspect of this work has been done before, so we'll be sort of following other people's footsteps in terms of the biological components. So we have here a muscle strip. This is from immortalized cell line. The muscle strips are made with an immortalized cell line. And the neurosphere, which I'll talk a little more about in a little bit, is derived from optogenetic mouse embryonic stem cells that we got from Roger Lab. And the swimmer, once we establish the co-culture and release it into fluid, so in this image in the right, this is the swimmer that's just freely floating in liquid. And when we actuate it by shining light, the neurons fire, muscles contract, and the swimmer moves forward very, very slowly, but still with a measurable speed. So this is sort of, taking the first step. This is just the first piece of the puzzle for integrating more complicated neural control. We also worked on a platform for co-culture. So this was here there are no, freely moving parts. The muscles are anchored to compliant pillars. The whole scaffold is just sitting in a petri dish, not going anywhere. This platform, we're still using the same cell sources, the same culture techniques for establishing the co-culture, same media formulations and everything, but it's just a platform where we can do the experiments more easily. Handling is easy. So we've used this to study a little bit in a little more depth what really happens when we co-culture these things. And in this particular design, we had 4 different muscles. So several things that we observed in the study before we shine any light, before we apply any stimulation to the neurons, the muscles are contracting. Now, this is not unusual if you culture muscles just by themselves, no neurons, just muscles alone. They can and often do just twitch by themselves. However, this is this spontaneous self-twitching of the muscles tends to be irregular, whereas the activity that we saw here, this is just a very short. While we were watching these samples under the microscope, they would twitch with a very regular frequency for a long time, like tens of minutes at a time. And when we shine light to stimulate the neurons, we get a more pronounced activity. So this is sort of an illustration of what we have. We have these muscle strips, which have individual muscle fibers, and a spheroid I'm calling it a neurosphere, a spheroid, which contains optogenetic motor neurons. And I'm not showing the data here, but we've done some tests to see that this activity that we're seeing, the cyclic twitching of the muscle, is most likely caused by firing of these neurons. So we think, we don't have direct evidence on this platform, but we have separate evidence of these cells cultured on MEAs that shows that these cells form networks that fire in rhythmic bursts. And they can develop these bursting patterns by themselves over time in culture. And the evolution of these patterns can be accelerated if we add muscle-derived or muscle-secreted soluble factors in the form of condition mediated. So there seems to be some synergistic interactions between muscles and neurons that result in enhanced outcomes in general. Now, after we did this work, I started thinking about what we want to do with the motile bots, with the biopods. We didn't want to just add motor neurons, because with the motor neurons, we still need some sort of external stimulation. Like with the swimmer, I still need to shine light to make it swim, or at least control its swimming. But I still need to do something. These are still not autonomous. And the whole point of adding integrating neuronal control is eventually at some point we want to be able to build these machines and do things by themselves, use their nervous system to maybe sense things in their environment and respond to them adaptively, maybe even have functions that resemble memory and learning. So I've been thinking recently and trying to read up on the neuroscience literature, developmental biology literature, how this actually works. So what we have here, initially my conception of what was going on with this within this neurosphere was akin to the old joke about the spherical cow. I'm from mechanical engineering background, so I'm prone to at first when I know nothing, I'm prone to coming up with generalized and simple and idealized models. But as and in this context, the spherical cow is basically the notion that these neuronal networks, let's say, are just all-purpose or general-purpose computing machines. They're a collection of neurons, and the neurons establish some sort of connectivity through synaptic connections or through gap junctions, most likely a combination of the two, and they generate these oscillatory patterns. Now, the more I kept reading, the more I realized that that's not really the case in nature. And by the way, these neurospheres that we have, they come from a mouse embryonic stem cell line, which was then transfected with chamaerodoxin by Sebastian Uzel from Saif Lab. And they have neurons, they have motor neurons, which we can identify by their HB9 GFP expression. But the people who have generated the original stem cell line have also shown that there are other types of neurons in these spheroids. And these spheroids are obtained by culturing the stem cells in suspension. The stem cells form aggregates called embroid bodies, spheroids, and then we stimulate the cells within these spheroids chemically by adding retinoic acid and morphamine or sodium cathode agonist. And these are morphogens that stimulate certain differentiation pathways. And we're trying to stimulate the cells, direct the cells towards differentiation into spinal cord motor neurons. And we do get motor neurons. These are the HB9 positive cells, but there are also other neurons that are not HB9 positive. They've shown that some of these neurons have known spinal interneuron identity based on their gene expression profiles. We also know that there are some glial cells in these populations, so it's a mixed population, and we don't exactly have a we don't know exactly how these cells are interacting and what kinds of circuits they're forming. So then I started thinking about, well, how does nature do it? How do these circuits work in nature? The circuits that control muscle activity. How do they work in nature? So most of what I've learned came from this review paper, an excellent review paper for anyone who's interested in this area. And in general, so Stan Gerlner here talks about a motor infrastructure, basically a hierarchical organization. So at the lowest level, at the cellular level, we have a single neuromuscular junction, or motor neuron, a muscle fiber that it innervates, and the interface between the two cells is the synapse. And then at the next level of the hierarchy, we have motor units. A motor unit is one motor neuron plus all the muscle fibers that it innervates. And the motor neurons themselves participate in these networks called pattern generators within the spinal cord. And then there's, as you go higher up, these circuits, this pattern generating circuits within the spinal cord, their activity is in turn modulated by signals coming higher up from the brain. So this image in the right, this is an illustration of the locomotory network of Lamprey, which is a kind of fish. With simple animal models like Lamprey, Drosophila, fruit fly, and zebrafish. So with simple vertebrate animal models, neuroscientists and developmental biologists have been able to identify these circuits. And sort of, it looks like there's a very well-conserved blueprint for these circuits. So in this particular diagram, each symbol represents a neuronal population, not a single cell, but a population of neurons, excitatory, inhibitory neurons, and a motor neuron. So this little rectangle here with the two excitatory neurons, two inhibitory neuronal populations, and the motor neuron population, this is just one CPG. And then there are many CPGs or segments that are organized along the length of the spinal cord, and then there's sensory feedback into these circuits, and the output of any one segment goes to the muscle through the motor neuron. In simpler animal models, these circuits become a little simpler. In mammals, for instance, these circuits become much more complicated. But in essence, it looks like there's a sort of footprint that's conserved. Now here's a really, really cool study. So this is from 1984. What they did here was they took a lamprey and they prepared it in such a way the animal is in a bath and is able to swim. And while it's swimming, they're recording electrical activity from the muscles. They're doing EM, electrophysiology activity of the muscles at these locations that are shown by the 1, 2, 3, and 4. And if you look at the electrical activity at these locations, as the animal swims, it swims by doing an undulatory movement of its whole body. And the muscle groups that are tagged here have a certain patterned activity with a certain frequency, and the different muscle groups, depending on their location, have a constant phase lag between them. So this phase lag illustrates the bending wave that goes from the head to tail. Now here's the really cool thing. What they did then is they excised the spinal cord and cultured it on a petri dish, in vitro or ex vivo. So if you isolate the spinal cord, culture it in vitro, and then do electrophysiological recordings from the segments of the spinal cord that correspond to the muscles that were measured here, you get pretty much the same activity. And this activity is in response to a global stimulation with glutamate, just adding glutamate in bath to the culture medium. So it's a global, non-specific stimulation. Yet the circuits in these spinal cords are organized such that kind of non-specific stimulation, global stimulation, can give rise to this. So that even the pattern of actuation itself and the coordination between different segments seems to be conserved. So overall, going back to the spherical cow thing, this notion that these are general purpose computing machines doesn't seem to work very well. Rather, it seems like these neural circuits, the neural infrastructure is an assembly of many, many very specific purpose computing machines. So going back to what we have, so this is a raster plot. This was work that we did with Saif Lab. This is data that Austin generated. And what we did was we took our stem cell neurospheres, dissociated them, plated the cells, which contain the motor neurons, interneurons, glial cells, and I don't know what else. So it's a mixed population. We plate them on a 2D MEA and record activity. So each row here is data from one electrode, and x-axis is the time. And we're seeing these pink boxes outline these bursts. So many electrodes are bursting all at the same time. So the whole network is bursting all at the same time, and they're bursting with a certain periodicity. Now going back to what happened in the Lamprey, it kind of looks like the pattern that is observed in one of these segments, one of these circuits. So maybe this might be a little far-fetched, but maybe what we have is close to what these individual pattern generating circuits. Now, we don't know that because we still don't know what it is, how it is that they generate these patterns, and we still have no idea if the phenotypic and genotypic specificity matches what is observed in vivo. So going forward, how do we actually make these nervous systems in vitro? How do we build muscle powered devices where the muscles are actuated by these coordinated activity of these assemblies of neural circuits? So in this image, this is from a study that was published in Cell a few years ago. So this is just one example of what is usually referred to as anatomical definition of these circuits. So in vivo, in nature, in animals, these circuits are highly specific. I've already mentioned their function seems to be conserved and very robust. Also, their morphology and their location within the body with respect to the rest of the body are also conserved. So in this particular case, the neurons, the motor neurons, the populations of neurons that innervate the foot, ankle, and hip muscles are located in these positions, in these anatomical positions, and even those positions are conserved. The different cell types here, they're referring to the V1 interneurons, there are also V2 interneurons, and the motor neurons, and the identity of these cells, their positions, all of these things seem to be conserved within a given species. So one of the challenges is how do we replicate this in vitro, and how do we replicate it in a reproducible manner? Now this is, strictly speaking, this is not about brain organoids. If anything, this is about spinal cord organoids, but I think this generalizes to, there are challenges that are faced in brain organoid, like cortical or hippocampal organoid area, that are very similar. How do we have this, how do we recapitulate the specificity in a reproducible manner? And if I want to build a biobot with multiple muscle groups that's able to do very complicated motions, then I would need multiple circuits. So how do we could put multiple spheroids, multiple organoids, but how do we, is it enough to just culture them together and allow them to interact, or do we need to have any engineering controls to allow that? And also, are we restricted by this evolutionary footprint? In other words, can we do it, nature did it this way, but can we do it in different ways? Yeah, so these are not rhetorical questions. I actually don't know the answers to these things. But I do think that trying to answer these questions is the proper way to go, or one of the proper ways to go about achieving successful integration of neuronal control into biopods. So I'll leave it at that, and